Welcome to the Slightly Educated Podcast. I'm Brandon Long. Today I'm talking with Howard Cromwell, who is a neuroscientist at BGSU. He does most of his research on the dopamine reward system, and there's a couple interesting things there that we talk about, mainly gambling and addiction, and why those are two problems that are very difficult to understand and interact with and solve. We get really in depth with the terminology, so I hope you don't shy away from this one because of that. One definition to keep in mind is the basal ganglia, which is a brain region that has to do with enabling and disabling movement. We also talk about ethology, which is interviewing animals in their own habitat rather than the lab. And of course we talk about frustrating rats and how exactly you do that in the lab. So all interesting stuff. Let's start. Okay, so I'm here with Howard Cromwell, who is a neuroscientist here at BGSU, and he mostly studies dopamine and reward systems, and that's mostly what we're going to talk about today. But if you want to just go ahead and briefly introduce yourself and talk about how you got into neuroscience.
Sure. So, Howard Casey Cromwell. I'm an associate professor here in Department of Psychology at BGSU, Bowling Green State University. I'm the director of what we've called now for about 10 years the Biology of Affect and Motivation Laboratory here at BGSU. And how I got into all this is quite a long story, but briefly, I got a PhD at University of Michigan in the biopsychology program, and really got hooked on the research at that point, and we were studying the brain basis of hedonics and pleasure using taste reactivity as a measure in the rodent model. And one of my earliest findings that I think is still pretty important is that these areas of hedonics are located in an area, or not located, but are heavily involved with a brain region called the basal ganglia. And as Brandon was saying, this area receives a flood of dopamine when we're experiencing something pleasurable. Now, my PhD advisor, though, at University of Michigan, really found out with some of his own research that these so-called pleasure signals with dopamine could also be reinterpreted as what he calls wanting signals, or signals of how can I get it and get it quickly and efficiently. And so his research, and I agree with, he's my advisor, of course, so I have to agree with his take on dopamine in that, from my earliest readings and work on this, that it is a compound that is heavily involved in this wanting signal. And so right away in my graduate studies, I was introduced to dopamine, pleasure, and wanting, and trying to figure out how all these things were interrelated. And then, so my background isn't real brief now, but I'm getting into some of the science on the way.
Yeah, go ahead.
I'll jump forward to another, what we do in our area is a postdoctoral fellowship that I stayed for about 3.5 to 4 years with a researcher over in Switzerland. His name is Dr. Wolfram Schultz, who, when I first started reading his work, also focused on dopamine signals in this special area of the brain called basal ganglia. And he talked about them as key regions of reward processing. And part of that is how we predict rewards. And Dr. Schultz's work was really focused, well, the majority, not all of it, but dopamine as a prediction signal. So I spent 3 or 4 years in his lab learning about the same system, the basal ganglia, the dopamine input into this area, and how it could be involved as a prediction signal, which seemed to make a lot of sense and seems to fit in some ways with the liking and the wanting as well. And then I'll skip other years, but then I arrived at Bowling Green State, set up my own lab, and we've been studying some of the same questions. I work with the rodent models mainly, and I couch the basal ganglia function in terms of a process I call incentive contrast. and think about it as a way that we compare rewards over time, and really focus on reward processing still. We typically don't do anything in our lab aversive to the animals. We like to have all the animals be happy, just like the graduate students and faculty. But they get a little bit frustrated and upset when just the reward level decreases or downshifts.
The graduate students? Or the...
No, the subjects. Well, the graduate students too. We all do. But you're right. So in our lab, we've been studying this reward downshift where they get more mildly frustrated. Sometimes we do get frustrated because we haven't frustrated them enough, but the animal subjects, and we try to see whether the brain signals, in particular areas in the basal ganglia, are encoding these comparable upshifts and downshifts. And part of my agenda is to examine whether the brain does this parametrically and consistently. And some of our data show that this might be the case. And so I can get into that in more detail, but we're really trying to drill down into how the brain makes these comparisons with between or among sets of rewards over time.
So it's relational, and so any reward is always compared to other rewards? Is that how it's...
Right, and we've been using magnitude as kind of the main upshift and downshift. And we food restrict the animals, so that's kind of important. And we can do that to different levels, but typically we've been clamping that so that all the subjects get food at a predictable time, and they kind of also know how much they're supposed to get. They work for these food items, and then still in a predictable fashion. We haven't really been mixing up non-predictability or lack of prediction in our scheme yet, but that would be something that we plan to look at in the future. But we basically, in a predictable fashion, do these upshifts and downshifts.
So when you say upshift and downshift, what is that exactly?
So with food magnitude, we use these tiny little food pellets. With some of my other research, we used to use more natural food items like pieces of banana or cheese or raisins and things like this. But right now, just for the sake of exact control, we use 45 milligram food pellets. We give them one food pellet, and we give that over a series of days, and then all of a sudden we'll give a cue that they already know about that increases food pellet magnitude, for example 3 or 5 pellets. And we see this elation effect or positive contrast. They'll work hard for the 5 pellets. We've been trying to identify some of their emotional indicators of how they actually are changing their emotional state after receiving the 5 pellets, and looking at a number of dependent measures, or what we call dependent measures, but behavioral indicators of greater motivation after they receive that. Now, the trick with incentive contrast is that we attempt to have a control condition, and this is usually within subject, where they get 5 pellets consistently in kind of an absolute, non-relational context, sort of a set of trials where you get 5 pellets. And we take, and this gets kind of crude in the behavioral sense, but we take a mean level of responding for their motivational indicators or their emotional state. And when that mean shifts, either with a single subject over trials or with a group of subjects over even a group of trials, then we say the rats are showing this positive contrast effect with 5 pellets. They're responding in a way when they're more motivated, more receiving possibly a greater hedonic reaction to that 5 pellets than they were when they were getting that without that relational component.
So upshifting is when they are getting more hedonic or more responsive to food, and then
Right.
downshifting is when they're less responsive?
Right,
Okay.
right. So we can do the reverse then, and you can't go below 1 typically from breaking up these pellets. So we would start an animal with 5 pellets consistently and then downshift them to 1. We've done a number of these, and we're not the only ones. There's actually a rich history of this using rodent models, and primate, turtle, honeybees. Lots of animals do this. So shifting from 10 down to 5, shifting from 5 to 1, shifting to 10 to 1. And that's kind of the parametric quality there. Part of my Because when, say, Dr. Schultz, who I worked with, or advisor at Michigan, or other people in the literature, they talk about reward processing as kind of this general ability of us to feel the reward almost in a dichotomous, yes or no state, and then not experience the reward. And so what we're trying to get into is again this: do you feel half as bad when you go from 10 to 5? Do you feel 10% as bad when you go from 10 to 1? And can you even get down, to lower levels of negative contrast as you go to a percentage under 1, for example? How sensitive are we to these shifts? And we can monitor that with our behavior, with measures of latency to the millisecond, and then we're trying to get this to the level of action potentials as well, and rate of action potential firing, or a different pattern, and trying to map that. So that's one of my major interests right now.
So you're looking at how these dopamine responses, when they go up or down, how they affect from the bottom up, like the action potential of the actual neuron, and then the behavior as well.
Exactly.
Okay.
Exactly.
So if I have this right, so you have like say a monkey in a lab, and
okay.
They know that when like a buzzer comes on, they can do a certain action and they'll get food.
Right.
What do you think? What do you take on the fact that like the anticipation is more rewarding in terms of like amount of dopamine in their system? There's
mm-hmm.
more of a dopamine response for the anticipation, right, than the reward. Is there?
So, according to the prediction theory, the dopamine signal changes. There's plasticity of the dopamine signal dependent upon the learning. So as a novice that comes into the situation, and it fits with the monkey because that's been one of the experimental subjects, but also with rat, and then there's even human neuroimaging to say this is pretty consistent with what happens in human as well. So there's really consistent data across species. That in the initial iterations, when we're learning, the dopamine is really linked to the reward itself. So this is typically a food item in the animal work, and then I know with some of the neuroimaging they're using some of the money, for example. A lot of times it's hypothetical money, but you've just won some jackpot. Yeah,
to a degree.
To a degree, you're right. Probably quantitatively it would be much bigger if you actually handed them the money right away. But then as the individual learns, so there can be so many different cues in the environment, but in these controlled laboratory situations, and with the animals I'm talking about, we have so much control. So we use tones, we can use lights, we can use complex visual stimuli, and they precede these reward outcomes. You can make this very predictable, and so let's create the most predictable environment we can. So we have 100% relationship between a cue and an outcome. We have something that's highly discriminable from anything the animal's ever experienced before, and bingo, you'll get a switch. Some of the data out there is shown from the Schultz lab that this can be within a single session, and you can watch that's kind of the fun experimental day when you're in there, just seeing the dopamine signal sort of just shift back to the cue and disappear to the reward itself.
So that occurs with learning. So like the more comfortable the animal is with knowing it will get the reward, the less dopamine for the reward, right?
The dopamine shifts. So the dopamine response basically decreases to the reward itself and becomes a potent response, time locked, or seen in the same time as the cue or this predictive event or object or whatever it is.
What do you see as being like the benefit of that happening, like from a biological, like evolutionary perspective? Like I guess why does the dopamine need to shift to the anticipation more than?
Well, the theory, one of the major ideas that this theory sort of moves into psychology is all about classical conditioning in some cases, how we actually end up using prediction in our lives, and in many ways how we're using prediction in a classical conditioning framework without us even being aware of what we're actually predicting. So dopamine can do this, of course, with a number of signals and cues in our lives that either we have as our habit or as we have as something that we could think of as a reflexive sort of response to these events and cues that are predicting things in our lives. And psychology for a long time, so all the way back to early notions of classical conditioning, has shown that without our ability to be able to predict, we're in big trouble. This isn't just our volitional, explicit knowledge of the world. These are our autonomic nervous system predictions for, what we want to eat, what we're going to digest. These are all the predictions throughout our body that allow us to prepare our body for good and bad things that are upcoming in the environment. So this is happening all the time. Psychology has emphasized this for a long time. So finding out that the dopamine is possibly a key signal of this is basically giving us the biological underpinnings of a key psychological process that is part of our everyday lives. And then, of course, a big part of the pathology of mental illness as well.
Yeah, it fits right in with the idea of you getting comfortable with a certain task and reward, and just getting kind of bored with it eventually, right?
Right. So in our real lives, yep, we can have 100% predictability, but it's quite rare. So typically in our real lives, these things are changing a lot. Having a whole realm of probabilities, but there are cases where we have boredom. We could imagine, I guess this area hasn't been studied as much as it could, but we could imagine the dopamine system going offline in a hypothetical world where everything is 100% predictive, and even the cues, the dopamine signal to the cues, eventually just fade away because in the learning prediction model, there's no learning yet to be had on this. In the experimental models and the work that's there, can easily show that the dopamine signal can get ramped back up when there's a new cue that predicts the old cue. And in the other case, when there's a chain of cues, it sort of keeps moving backward in time to allow for greater and greater prediction that's earlier and earlier in time.
Okay.
And so that's probably continually happening. And then the other experimental manipulation is that they take away the 100% and see how much of the dopamine is left when you start to vary things between 0 and 100%, and seeing that in a more real-life case scenario, the dopamine signal is perpetuated in time, always trying to figure out, is the predictive quality of this, how much is it changing, and how much dopamine do I need to keep this going as a learning signal? This is something that's a valid predictive signal.
So you're saying dopamine is kind of a driver of learning predictive behaviors to kind of exploit the environment, you could say.
Right. Yeah.
It's kind of, you were just talking about unpredictability, and don't you see that when you have there's like an animal does a certain action to get a reward, and there's like a 50% chance that it'll get a pellet or not. Do you see higher spikes of dopamine? Is that a response to try to figure out how to get the reward more predictably? Would that be a good analysis of that, or?
Yeah, so 50-50 is tricky then. So we can imagine the subject actually understanding the 50-50 or flipping a coin. And in some ways, with humans, for example, we could imagine that we have this knowledge, actually, because someone's told us, for example, that there's no ability to predict in this particular case. Hypothetically, dopamine should basically not be playing a role in many ways, right? So that's a reward prediction error hypothesis in some ways. It would be giving us the error when if 50% no reward, and then a signal 50% reward, but in hopes of gaining information here that there's something beyond 50-50. Once we're aware of that, the dopamine signal is basically saying that being able to predict, it's not going to be playing a role. Now, in human and animal models, it's not necessarily the case. It's almost as if the dopamine signal kind of goes up and goes back down. It goes back up, goes back down. And in the animal model case, it's very difficult to embed this kind of 50-50 knowledge. In some ways that the animals are always either pessimistic or optimistic and playing a larger role of their stochastic kind of probabilities and trying to figure out, is there some movement away from 50-50.
Yeah, because even with gambling, you can fully explain to them, and they probably know how dismal their
Right.
odds are, but they
Right.
still partake in it.
And so we have these natural tendencies, and so we could think about the dopamine system embedded into either these primal or instinctual tendencies to and embed within the personality that we have a natural bent toward loss aversion and being risk averse and personality types here, hedging our bets that it's going to be less than 50 or low probability based on the way they read the environment, and then other individuals through experience all of a sudden being risk prone, and I don't know if you want to say seeking loss, because that doesn't make sense, but I mean it doesn't make sense in many ways, but then when we look at pathological gambling we point to that and we go, yeah, that's it. What's their dopamine system like? And that is a major area of research. Actually, we're getting into that a little bit as well, kind of with our incentive contrast in ways that we think animals are insensitive to contrast. They're not paying attention to the comparisons. And so we've tried to look at particular animal models or animals with particular experiences, seeing how it changes their ways that they should naturally be tuned into these upshifts and downshifts and the ways that they should respond, either be more motivated or give up. And it's a major area of research. And you might want to know answers like, well, how does the dopamine system change in pathological gambling? I think this is one of those answers I have to say to be determined. There's definitely a way to say that they seem to be hypersensitive to reward in some cases. They seem to be exaggerated responses to wins, and sort of a pathological or atypical way that they're altered in their predictability, looking at 50-50s or below 50% as something they're learning about or finding rewarding. So those kinds of findings are there, but they're tricky to study, and animal models of gambling are hard. We've actually been trying to do it in the lab, and one of my take-home messages on that?
What's that?
That most of the animals are too smart to gamble.
Really?
So, they're just very risk averse. When they get zero, they don't go back to that outcome.
So you have to be as smart as a primate to be stupid enough to gamble?
Even a rat. So talking about some of the rat models we've been doing recently. So, it's hard to study then in our controlled environments. What people do are stress the animal models, have a developmental cascade of events they're exposed to, and so it's really tricky in that way.
And you want to kind of figure out the age-old question is like, did the gambler choose to gamble, or was the gambler already predisposed to gamble? And
right.
Yeah, that's an interesting area.
In general addiction, and usually pharmacological addiction, there's some evidence to show that individuals with high risk, that is with family members who have been diagnosed with addiction disorders, show different brain profiles. The one related to dopamine, for example, that's still in our textbooks, and it's been validated with several studies, is that individuals have actually a ramped down dopamine system. That some of their prior to showing their addictive behavior, they seem to have fewer receptors, for example, for dopamine in some of these regions like the basal ganglia and the forebrain. And so part of the explanation has been from this prospective imaging research, for example, that these high-risk individuals have a greater need for stimulation. This is kind of the sensation-seeking answer of a personality type, that they go down that pathway with the particular experiences because their brain needs that. It needs that 50-50 or less to get excited.
Have you done any looking into how context plays into dopamine reward and I guess it's been looked into a lot in addiction. Like
Yes.
people used to like drink in a certain place, and
Right.
they go back in that setting, not necessarily even like other people like egging them on, but like just the certain situation or place, location, puts back in them the want to go back into those bad habits.
Right. Right. So in the field called psychopharmacology, and a lot of the work again using animal models, looking at things like conditioned tolerance, for example, where an individual is more likely to survive in some cases, but be able to handle physiologically a dose of a drug because they're in a certain location, because they've had experience with that drug, with these paired with these cues, and some of the research points back to the dopamine signal here, and points back to the predictive cues that the dopamine is working through a network in the basal ganglia and other regions in the brain to prepare the body to be able to handle that drug dose. This is kind of brain and body then whole physiological response that enables the individual to, if you just took the injection of the heroin as an example, it's clearly been shown, and there's just so many cases of this from the human anecdotes, I guess, from the human addicts. But the animal models clearly show that the specific cues can be powerful, that the dopamine signals that enable that are part of the response to the cues. But it's more than that, of course. It's probably a host of neurochemicals that are involved in this conditioned tolerance, and then the other flip side, or sort of conditioned high. It's not, in my mind, from what I've read, not as much research on that. It's almost as if if you take that to an extreme in your mind, you could almost go into a location and then not need your drug. This is kind of a exaggerated take on what conditioned high might be. You have the cues and they actually replace the hedonics or the euphoric reaction of the drug. This has been another area of research. It also involves dopamine, but other compounds. I guess you could throw out another neurochemical that we call the endogenous opiates, like enkephalin and endorphin, that can be released in response to particular cues, and might give someone, for example, this natural high or a conditioned high. And so those avenues of research, I think, are really exciting in reward processing. To me, they involve prediction. They also involve an individual just retrieving a memory, I think. That's where I've kind of gained at with incentive contrast as well. Just not that we need to manipulate prediction, but we need to understand how much these cues, what the kind of representations of those cues are in terms of how much of a rewarding response in relation to another cue or another experience. So, and some of my colleagues have said that I'm studying something that is probably key to the whole phenomenon of reward processing and drug addiction and so on. But it's so complex that we may not even have the neural ways to manipulate or monitor this kind of, and with some of our data I almost agree, because I see some of the effects, but we haven't really been able to find the parametric kind of relationship that I've been looking for yet. I keep thinking we don't have the right behavioral paradigm yet to really measure.
Because certainly, you can get lab animals to just not even participate in some like reward games if you inhibit their dopamine. So it seems like the answer is somewhere there to fix addiction, but exactly where is anyone's guess.
Right. Pharmaceutical manipulations are definitely at the forefront right now, but they seem to be kind of gross, kind of primitive, in a way that we know that the brain is so much more delicate and and precise in the way that they're handling this information. And so that's going to be a trick in the future, I think.
So something I ask all scientists usually is that, so do you partake in the science because it's something that is gratifying itself, or is it you're just trying to discover new, I guess you could say, truths, or do you like what it can do for like humanity as a whole?
Well, yeah, I'd like to hear some of the other answers, because I think people have to answer honestly. They have to answer both. I think sometimes you start out with the furtherance of knowledge and your own basic kind of research interests, like looking back as an early student and learning about it. At least in my mind, I think, unless some individuals, I guess, have more of an experience with particular illnesses and get involved in the medical side. But for me, at least, I was involved in the basic research and really learning from classroom settings about the mystery of the brain. And so I guess the first question was, in the classroom, how does the brain work? And then when you get done with the class, you're sort of saying, well, we didn't answer that question.
Yeah.
And so I'm still doing that. I start the class, and then we end and we don't know. And so, I told some colleagues as well, isn't this kind of ridiculous? But, we won't retire until we figure out how the brain works. I think this neural relativity is a key component that's been underlooked, under examined. So I think we could make huge breakthroughs with what I'm calling, there isn't really that term out there so much, but neural relativity and the reward side of that.
So how it interacts with everything, the environment and the whole slew of... But
doing it in a well-controlled manner so that we know what the relative context is, and then we compare it to an absolute context in the lab. And so I think that's one of the things that the brain does and how it works, at least within the psychological way. It's been not explored in a lot of detail. And then I remember my advisor coming in graduate school, he said, well, we're going to be working in the basal ganglia system. I said, well, what does that do? He said, look up Parkinson and Huntington's disease. When dopamine is gone and the signal isn't there, the basal ganglia, people don't move. They move slowly first, and then they stop moving altogether, and they become rigid. And we were studying Parkinson models, really, and movement sequencing with basal ganglia. But what ended up really just overwhelming this research was figuring out the same brain region has all these reward signals to it, and this reward expectancy signal, which you've been talking about and we've been thinking about, that is just really cool when you're looking at it, because when you play the cue and you're listening to the neuron, it just begins with a cue, and then it just continues as neural firing until the reward is obtained. And this can be over a matter of seconds, and we've even sort of prolonged that time period, for tens of seconds. And it's as if this neuron sees something, hears something, experiences something, and just stays active until it gets what it wants. And to me, it just didn't fit into the Parkinson's and Huntington's scheme for basal ganglia. So part of my PhD work, and the title of my dissertation was Functional Heterogeneity of this Basal Ganglia System, was that there are these motor subsets, regions that are critical for movement onset, movement termination, movement sequencing, but then there's this reward processing sub-areas. And when we looked into Parkinson's and Huntington's, we found out these reward deficits. So I've been talking with some students just this past week on gambling disorder in Parkinson's patients when they start to take their dopamine elevating treatments, and seeing that the rise of pathological gambling is actually significantly higher, especially with early stage Parkinson's patients, than anyone would ever expect. So that's sort of where you have this motor neurological disorder, and then all of a sudden their treatments impacting their reward processing ability.
Do you want to talk about, I don't know if you have an answer for this, but what's like the most profound thing that you've discovered in your research, if you have anything about that?
I think I mean, I think one of our recent findings, I just think it was a really neat study using the rat model. We ended up developing a naturalistic environment up in our laboratory. It's about 10 to 12 feet long that we put the animals in. And to me, it's one of the first cases where we've been studying the animals in an environment where we actually have them run around the environment in different locations to get different rewards. It's so hard to do with other animal models, but in the rat we sort of have this naturalistic little tube-like environment. And I call it the self-pacing, sort of free-foraging choice environment. We're trying to think of a shorter name. It's something fun.
Yeah, but I don't
Yeah.
you make the titles of the papers anymore if you're going to name it that long.
I know. They're all, yeah. And we ran the animals over a series of weeks, and we ended up deciding that their free choice is divided up into different reward abilities. And they can be separable. So we have their ability to discriminate between the two reward outcomes. And we can show that they can easily do this, but then if we make them harder, they start to generalize and show, more equalized choice. But then once they discriminate, we think that that's necessary, though, to move into abilities to make preferences. So they have to be able to make the difference there. And they do that quite well in most scenarios. And then once they can do that, and they do that the best, they start to show a preference. And then with these two decision-making qualities in the reward process, they also impact, and we were studying at the same time across weeks, this incentive contrast ability. And we showed that when they're showing the most optimal choice, getting the most food over the rate of time, all three of these, discrimination, preference, and incentive contrast, are working together and optimizing. But we showed when we made the outcomes more difficult and discrimination broke down, so did preference and incentive contrast. And then we made a perturbation in this area of the basal ganglia and showed that all three of these could be altered, so that this area in the brain seems to be integrating information at different levels, taking the magnitude between two rewards as something important, taking an experience with preference and placing this on top to make what we call a value hierarchy. And then once the value hierarchy is there, being able to take this and use your experience with what I just had on that hierarchy to make a decision about whether this value ranking is appropriate or not, and even shifting up and down on the hierarchy depending upon. And that's real complicated, I know, and maybe it's one of the reasons some of my best findings aren't as popular as they are right now, because we're still trying to figure out how to say this in a 30-second time bit. But we called the title of the paper is Fractionating Free Choice. And to me it's kind of groundbreaking because it does divide choice into these different kinds of what I call reward processing components. Start to talk about how they're interrelated, and start to talk about how a key region in the brain, it's a striatum actually, it's not the dopamine cells per se, but the striatum receives dopamine. We're trying to look at how dopamine is a key player in this, but how these neurons in the striatum are integrating the information to optimize decision making and choice.
Okay.
That's a recent, it's 2 or 3 years old, but I thought it's really cool. It's caught my super interest. It's caught some interest out there in neuroscience right now.
So it's complex. You can't.
It's too complex. Yeah. And so sometimes we just have to realize the brain is complex, and that a lot of the sound bites for these are going to be more than a few seconds. They're going to be a couple of minutes to realize the importance of it.
Yeah. It seems like with that study you were trying to get. That was one of my questions. Was what do you think about the field that is more trying to get rats in their natural environment rather than because like a big critique is that you noticed a 30% shrinkage in this brain region when you did this when this happened in this rat instead of this rat.
Yeah.
But if you look at the actual rat world, like it's like 70% bigger than pretty much both of the rats.
It's a great point. Thanks for bringing that up. I could just lecture on that forever. I'm a big fan of ethology. Ethology in the ethological tradition is observation of animals in the natural world. Do I actually do that or use it? No, but I try in the lab settings to do that. So this was one case in point We also have a rich history here at Bowling Green State University. I have to mention another researcher's name just then for all of this posterity, Dr. Jaak Panksepp. I don't know if that name rings a bell for you, but he was a researcher here at BGSU for about 25 years. And he advocated that animals have the same primal basic emotions. All animals share these, so rats and humans, they have the same basic emotions. And we have the same brain substrates that are producing these basic primal emotions. Humans have an encephalization that can change how these emotions operate, but we still at the root of our brain, and really did focus on the brain stem, I guess it's not the root then, where these primal basic emotions are. But he kept trying over and over and over again to study more naturalistic behaviors. Case in point, play in rats, and he was one of the first researchers to look at rat play. They only do this for about 2 or 3 weeks of their lives, well, maybe 3 or 4, about the first month, a month and a half. So you have to catch them when they're young, little juvenile rats, put them together with litter mates, and they'll jump on each other and tackle, and it looks like wrestling and aggression, but they'll take turns and tap on each other's shoulders. Do rats have shoulders? Or the little area there?
Yeah,
they
do.
They do.
Pretty tiny, but...
yeah. So he focused on that, and he said that if you look in a burrow environment, visible burrow, if you look at some of the rat models, they play. And then, of course, other people picked this up, and there was already a sort of ecological literature on this, but animal play in the wild has been kind of a neat area of research, but doing it in the lab. Then he looked at neurochemistry of play and the brain basis of play. I worked with him a little bit while he was still here. He passed away about 1.5 years ago, Dr. Panksepp. Another case in point is, and we did a project on this too, talking about using a naturalistic stimuli. He said, "Why don't other people have brought a cat into the lab? What are rats afraid of? Cats or dogs? But to make it more systematic, instead of the whole cat, we clipped cat hair and put cat hair in a little vial that then would emanate, or the odor would disseminate from the container." Panksepp published 20 years ago, you put the cat hair in a lab rat's cage and they just freeze. They won't move. You put it in the juvenile rat's cage where they've actually experienced play or in the midst of their play, and they just stop. So they have an instinctual, hardwired program.
Yeah.
These are lab rats bred in these distributors that have never experienced a cat, for sure. And their response to that, I don't think I've ever seen anything so dramatic in an animal model before.
Yeah, those are very interesting, because in some cases you have scenarios where humans they've learned negative associations with I think it's snakes very quickly, but it's not innate like with the rats. With the rats, it's just there.
Yeah,
that's a whole nother.
What is the unconditioned response of fear in human? It is tricky. It's actually surprised me over the years to think about what we're scared of, or what we should be scared of. I bring this up in some classes. We've actually watched the video of John Watson's work with little Albert. There are some videos online you can see on this. I was always struck that when he's doing his classical conditioning experience with this little Albert, that Albert's not afraid of the animals that he puts down in front of Albert until he pairs it with a loud noise behind Albert's head. That was his classical conditioning. So loud noises, for sure. We're all born with this kind of parachute-like movement there when we hear a big bang behind our head. Also, I think looming stimuli have been looked at in young infants, and consistently we have this kind of autonomic fear response, dilation of the pupils at least. But yeah, sometimes other than that, we would think that like a baby rat, we should automatically, reflexively be scared of. But there are some things, and so that's where Panksepp says there's a link, and that we do have unconditioned response fear network, and then it quickly, for humans, probably very quickly, gets linked to conditioned cues, and on the positive side of things, dopamine can take over. On the negative side, Panksepp studied a whole network of fear areas, and there's so many different researchers out there who are looking at this, of course.
Well, that's about all we have. That's right about
Okay.
45. Again, thanks for your time.